We're joined here this morning by Michael Roberts, who is a life science entrepreneur and has had a very successful period with Synpromics, started a company there and had a successful exit to Asclepius and is now doing his new thing, which is Chromatin Bioscience. So welcome, Michael.
Hello. Hi there, Ian. How are you doing?
Not too bad at all, thanks. Not too bad. So, I suppose let's just get into it. Tell me a bit about your background and your upbringing, because it's always interesting to find, where some of our entrepreneurs have actually come from.
Sure. So I was born in Edinburgh, but when I was fairly small, we moved out to West Lothian. So I spent quite a lot of my kind of former years out there. Really had an outdoorsy life, out all the time, playing, what have you. Then we moved back to Edinburgh, just in time to go to high school. And I went to a school called St Augustine's. I would say that there was no pressure at home to kind of go into university or anything like that. But, from quite an early age, I wanted to be a medic. So I kind of had my mind on going to university. And I actually worked Saturday jobs and that sort of thing to get money so I could pay for tuition to help me in subjects that I wasn't that great at. So I don't know how many of my other kind of fellow classmates were doing something similar, but I did actually manage to kind of pass my hires, got a place at Glasgow Uni, and I was actually awarded an award from school as the pupil who was probably least expected to get into uni, or at least the people that did better than expected.
Did you go to Glasgow University to do medicine then?
No, my grades weren't good enough. So what had happened when I was at school, I was doing all the sciences, and there wasn't really the timetable didn't allow for pupils to do all three sciences: biology, chemistry, and physics. So what the school let me do for my standard grade during the lunch times was to kind of go and copy the biology notes and basically study for the exam on my own.
Really?
So that, I think, really got me into biology because it was my own responsibility to do it. And I had to kind of learn by myself. I mean, the teacher was available, but he wasn't that happy all the time to kind of answer my questions in the middle of his lunch break. But I did well, passed the exam and got into hires. And I think it was that made me more interested in biology than medicine. So I applied to biochemistry.
Did your parents have any interest in the kind of biochemistry or life sciences kind of market or whatever in any way, sort of shape or form?
No, not really. I mean, I was the first person from my family, from my extended family, to go to uni. So like I mentioned before, there was no pressure, and I think they just let me get on with it. And, so there was no prior interest. It was just, when I was a kid, I was always had kind of microscopes and science kits and all that sort of thing. But it was very much off my own back.
On your own volition. Yeah, okay. And in terms, I suppose, of the moment at which you kind of decided to do biochemistry, was there a kind of external inspiration, or did the inspiration really come from the study?
It came from the study in gene regulation in particular. Right. I remember when I first kind of, understood what a gene was. That was really, really interesting. So, I mean, the way that Glasgow University worked at the time is your first year was all the sciences. So again, I did chemistry, physics, and biology in the first year. So you still have the option to kind of change. So I was kind of definitely interested. I could have gone into chemistry or physics because I found physics really interesting also.
Yeah.
But by the time I got to second year, I was just excelling it in genetics, molecular biology, biochemistry. Whereas at school, I would be lucky if I got a B grade. I was typically getting 80s and 90% at uni because I just found the subject matter really interesting.
Right. As all began to click, and I take it the fascination just grew.
Yeah. I mean, I would say as an undergraduate, that's when I really got into gene regulation. Definitely. And I remember one of the exam questions they had was, if you were to design a conference to talk about gene regulation, what would you have in that conference? What would the agenda be? And that was one of the questions that I answered, and my mates were like, why on earth did you answer that question? But it's just, I was really interested about, how genes are controlled and how transcription happens. And at that time, there was a debate around methylation, whether methylation is, what happens first, is the chromatin open and then it's methylated, or is it the other way around? So I found that really interesting. And by the end, I got a first class degree, and that really kind of opened up my options.
Where did you go? Do your PhD? Were you at Glasgow University as well?
No, I went to Cambridge, so I got offers. I got an offer from Oxford to do a PhD in immunology, and then an offer from Cambridge to do neuroscience. And I took the Cambridge offer because it was delivering genes into neurons using viruses. I thought that was really cool. And even at that stage, before I started the PhD, I thought that would have great applications in gene therapy, which is obviously what gene therapy was at the time.
Yeah.
So it was like a no-brainer for me. I was going to do that project. And that kind of opened up a lot of other opportunities as well.
Yeah, certainly sounds like it did.
I mean, it wasn't all plain sailing doing the PhD. I got into the social life quite a bit there, and I was rowing for the college first aid, and and my supervisor after a year basically sat me down and said, "Are you sure you want to do a PhD?" So that kind of gave me a kick up the ass to actually, maybe I should finish this off. But it's a really difficult project, because there's a lot of cloning involved. Now, of course, you do gene synthesis, but there's a lot of difficult cloning, and I really struggled with that at the beginning. But some of the people who'd actually been responsible for kind of the genetic engineering revolution in the 70s were at Cambridge, so my supervisor packed me off to their labs. Right. I guess I learned to clone from the people that invented the science, as it were, and from then it became kind of one of my strengths.
Right. Wow.
I kind of pieces of DNA.
Yeah. Clearly steeped in academia up to a point, slightly kind of distracted perhaps by some of the social life and so on. But at what point did you decide that academia wasn't for you, and that the kind of business world and becoming an entrepreneur was something that was attractive?
Yeah, so after my PhD, I did a postdoc in London, Roy Holloway, with a guy called George Dixon, who was quite a strong name in gene therapy of neuromuscular disorders. So I did that kind of work with him for 4 years, and that was quite a productive period from an academic perspective in terms of publications. And then I saw an opportunity with Marie Curie to go to Greece. So I thought I'll do that. And the reason that I got interested in that, again, it comes back to gene regulation. This is back in 2002, and microarrays had just come out as a technology, and I just found it really interesting to kind of study what thousands of genes do at any one time in a particular cell. So this was a project in cancer, so it's a little bit away from neuroscience and neuromuscular disease, which I've kind of been involved in previously. I just loved the concept of going to Greece and setting up a microarray in the facility at the institute. So that was supposed to be 2 years. Again, it was still an academic role. And I ended up staying in Greece for 10 years after that. But after the fellowship had stopped, then there was an opportunity to work at a small biotech company, which was founded out of Mountain View in California. It was called Regulon. And the CEO was Greek, and he was running his clinical trials in Athens. And that's why he had labs there. So that role was to basically apply an element of gene therapy to his platform. So he had a non-viral liposomal delivery platform, and he was using it to encapsulate platinum compounds in order to improve chemotherapy to kind of reduce the side effects of cisplatin. So I was recruited there to run the molecular biology lab. I was only the fourth employee, and then he said to me that you've got a trustworthy face. Go off to BioEurope and kind of see what licensing deals you can get for the drugs that he was developing. And that really got me interested. So I did a few of those events, and yeah, I just liked it. I just liked going there and kind of pitching applicable science to various companies. And then that just kind of sparked an interest in corporate strategy and really all aspects of running a business. So eventually I got on the board of that company, and then I decided, I'd had this idea on gene regulation for some time. So after about 5 or 6 years, I decided, actually, I'm going to kind of go back to the UK and go for this company, this idea that I have.
Right. So that was going to be my next question, actually. What sparked the idea behind Synpromics and why was it set up? But, clearly you've come to that. So in the early days, I suppose moving on to the next bit, what was the biggest challenge in establishing Synpromics? So,
I think the biggest challenge is getting the right team together, the leadership team particularly. So our first project at Synpromics was with an agricultural company, and it was Dow AgroSciences. And we were introduced to Dow Ventures by Scottish Enterprise, who'd hosted them in Falkirk. So we went to Falkirk House, and I think they were more interested in chemistry ideas. So I was the only biologist in a room full of companies that kind of had various kind of chemistry ideas. And I pitched the idea behind gene regulation to Dow Ventures, and they really liked it. They went internally. Then Dow AgroSciences kind of got interested. We went out to the US, pitched the idea there, and they ended up putting money into the company so that we could make them promoters in corn. And this was completely far away from my background, agriculture and what have you. I mean, the idea behind Synpromics came very much from a gene therapy perspective, where I'd just come off a gene therapy postdoc, gone into genomics, and I'd read a paper about synthetic promoters where they'd taken the transcription factor binding sites and shuffled these in muscle and created some really neat promoters, and I thought it was a great idea. And then I immediately saw the application of data from genomics analysis. You could actually use genomics analysis to find parts to build these promoter libraries. So that was kind of the idea behind Synpromics, and that's what I went out pitching. But that was from gene therapy. So to kind of have the first customer wanting to use that for plants was a bit of a challenge, I would say. So, and also getting the team together. So our first advisors and all that, they were all kind of professors in plant sciences. So that was quite a challenge getting a team together.
So kind of simply put, you had sold something in a sense, and then had to go and put the business together that would actually kind of deliver on it.
Yeah, exactly.
Very good. Right, okay. Good way around.
So, I mean, but it's what I learned later with various courses that I went on. It should always be customer driven. And I mean, even I was on a course recently for another company that I'm helping, and I mean, that was great advice that the person that was giving there. He said, even if you don't have the finished product, you don't have to let your customer know that you don't have the finished product. The finished product is what you're going to deliver for them, and it's very true.
Yep. Over perfection, or continually kind of trying to reach that point where you've got a perfect product is a difficult challenge, and I suppose especially for folks who have got a very clear view of what that should be. Perhaps approaching customers with something that's not complete and needs development and so on, and but still believing in it enough, and creating enough passion within the customer and so on to get the business rolling. It can be quite tough, I would imagine.
It was funny because I pitched to a bunch of plant scientists promoter library screening in colorectal cancer cells, and they still went for it. So,
yeah, well, the powers of persuasion.
And then later, when we went into gene therapy, we pitched all this promoter library screening in maize to a whole bunch of gene therapy people and investors. So it is power of persuasion, and being lucky enough that the person that you're pitching to can see the potential.
Yeah, absolutely. So the building the team was a tough aspect, I suppose, in the early stages. Bringing that all together, getting yourself into a situation where you had a proper company, and going from there. What then after that had kind of happened? I mean, I'm brushing over what was probably a pretty tough kind of period in some ways, but once you'd had the kind of established team of folks to take the business forward, what then became your biggest challenge?
I think delivering on projects, because we were custom designing projects, and we're making promoters, and until that point the industry had been using viral promoters in order to drive expression of genes in gene therapy. And viral promoters work very plug and play. You could put them almost anywhere next to a gene, and they're going to make the gene, they're going to express the gene into protein.
Right.
That's not necessarily true with promoters derived from the human genome. You have to really put them in you have to really think about where you're going to put them, what's the intervening sequence between the promoter and the gene. So that caused some scary moments at times, because we would screen different promoters using fluorescent protein in order to kind of find the best combination of parts. Then you'd put the gene of interest next to the promoter, and it wouldn't work anywhere near as well as the fluorescent protein. And that kind of brought a little bit of doubt, is the platform going to work, and etc. But after the initial shock, you soon learned that you just have to kind of think about what you're going to put down, or what sequences are immediately adjacent to the promoter, and you can do that by you can look at the effect of different sequences very easy with different biochemical assays. So once you kind of start getting more involved into that, you realize that it isn't actually a problem. You just have to be careful as to how you make the design of the promoter with the gene and other aspects.
Right.
But I would say that caused some sleepless nights in terms of how we're going to deliver on our projects if the promoters aren't kind of working as well as control promoters when you shoot them with a gene of interest.
Yeah.
But we got around that, and I think this year, one of the companies that we worked with, Unicure, so one of the early projects is now going into the clinic. So so that's been really, really good. So
very exciting, very rewarding. I'm assuming, seeing that come to completion. What then, I suppose, did you do? And you've kind of answered this in a way as well. But what did you do to commercialize the business further? Beyond obviously initial conversations with Dow and so on. How did you go about kind of, I suppose, engaging the broader market?
Oh, that was all kind of these BD events. So when we made the switch from agriculture to cell and gene therapy, and that also was coincided with bringing on David Venables as CEO and John Brown as chairman. So that, we really had a great team there. So it was just kind of going out with those guys and going through their contacts, going to meetings like the American Society of Gene Therapy, Bio Europe, Bio, kind of JP Morgan, all those sorts of things, and just doing a really intense BD exercise.
Yeah.
So that at one point, we had over 35 partnerships. And that was a mix, mainly gene therapy projects, but also bioprocessing, biologics manufacturing projects as well. So making promoters that increase the yields from CHO cells and that sort of thing.
Yep.
So, I mean, at one point we were working with 6 of the top 10 publicly listed gene therapy companies in the US. We were working with organizations like Lonza, GE Healthcare, Sartorius, Oxford Biomedica. So just kind of getting the technology out there, presenting at academic conferences, and going to partnering events.
Right. Were you all involved in that, you and David and the chairman, or how did you organise it?
Well, John is a vast network. So he would go to different events, but not necessarily in the capacity as our chairman. He was also the chairman of the Cell and Gene Therapy Catapult.
Right.
So he would facilitate introductions, but it would be David and I out at these events, basically.
I'm assuming then that those pitches were very valuable in terms of building those top-end clients that you've mentioned there. Yeah,
absolutely. I mean, I think this is still the case with gene therapy, but there are two issues. One is delivery. How do you get your gene into the target's organ or cell type? And to a lesser extent, it's how do you control the gene when you get there?
Yeah.
So because delivery is such an issue, there's an awful lot of research activity, and a lot of companies are kind of building these delivery systems. So it was recognized that delivery was an issue, and then with Synpromics, we were really pushing also that once you got the delivery, gene control is also going to be the major issue, because you can't use a viral promoter because they're uncontrollable. They just, they might go on forever, or they might just switch off as well. So they're unreliable. And we got a lot of traction. People then really thought, we really need to think about gene control as well. So I think we were lucky in that people were developing these platforms, particularly in AAV, to improve gene delivery. And we're beginning to solve that so that they could then turn their attention to controlling the cargo when it gets into the cell.
Right.
So there was an appetite. And also from a funding perspective, we got a lot of money from Innovate UK and Scottish Enterprise, for that matter. Because the UK government had established synthetic biology as one of the 8 great technologies. So they had a lot of grant calls for synthetic biology. And that just came at the right time for us to kind of validate the platform. So not only were we doing cell and gene therapy projects, but we were also doing projects in yeast.
Right.
For instance.
Okay. So a bit of luck at play there, but clearly, I suppose it's always one of these kind of processes of you working hard and getting lucky at the same time, and the market being ready for it, and so on.
Exactly.
That played in your hands. Looking then more, I suppose, at the kind of corporate structure, your investors, the journey that you've been on with them. What advice would you give to others who are in a similar situation, basically trying to build a company with investors and I suppose the broader interested parties in your organisation? How did you do that, and what advice would you give?
The best advice I can give there, I think, is work with investors that trust you. Now, typically when an investor looks at a business, they look at the idea and then they look at the team. And it's obviously, the team's very important, and some probably more important than the idea in certain instances. So I think it's really building or finding investors that you can feel that there's going to be trust there. Not necessarily, it's not just you trusting them, but making sure that they can trust your judgment. And making sure you have a team in place that they can have full confidence in, because there will be decisions that have to be made down the line around the board table where everyone's not going to agree as to the best course of action. So basically you want a leadership team in place where the board and the investors trust the team to make the right decisions. And because if you don't have that, then it's going to be really kind of difficult in order to do anything, basically. We were lucky with our investors. We had we didn't have a huge number of different parties. We had Calculus Capital and the Scottish Investment Bank.
Right.
And Calculus basically let us go on with it, I would say. And we created a great board full of experienced people. SIB were even lighter touch than Calculus, because I think their model is to kind of lean on the corporate venture partner.
Right. Okay.
And I think when it came to the exit, there were a number of different opportunities that presented themselves, and we ended up with a cash exit. But it's decisions like that, when you get to that point, that you really need everybody to be aligned on board. So that's what I mean. Just make sure you kind of build a team that can be trusted.
And is that something that you can kind of build in earlier in the process, so that, I don't know, there's a structure by the time you get to, I suppose, the exit point that would follow something that everybody has already pretty much agreed with? Or, is it more difficult once you reach that point to kind of, I don't know, herd everybody in the same direction, because maybe some of their kind of views on the value or whatever are different?
Yeah, I think you need to work with people with an understanding of the space, and you need to have an early discussion, I think, with what exit's going to look like. Because there's multiple different, you could go IPO, you could go acquisition, you could go complete, do merger.
Yep.
So I think it's good to have discuss those options at the board level fairly early on, in order so that the board knows kind of what to expect or what people's views are.
Yep.
With different scenarios. And that's something we didn't do.
Yeah.
But I would say that was something that I would kind of, and it's, you might think it's premature to talk about exit fairly early on, but at least if you have an understanding what everyone's views are, it doesn't mean you have to go and execute on it. It's just, where people stand, and you might want to have it as a regular agenda item every year or during the strategic discussion. So, yeah. Yeah,
it makes a lot of sense. I mean, I think a lawyer that I worked with a long, long time ago once gave me, some very good advice about sorting out, simple things like, your memorandum and articles and so on and so forth, so that and we were shareholder directors and so on, so that we were all aligned. But of course, we completely ignored that advice and cracked on regardless. Yeah. Only to stumble over it a number of years later, which we were fortunate to kind of get through. But, I think if we had, set things out in a way that was, much more kind of aligned and understood early on, it would have been an awful lot easier. But it's a bit like signing a prenup in some instances, isn't it? There's a sense that it's binding you, and that I don't trust you from the outset, and maybe.
Well, we had a great experience with our investors. I mean, they did let us get on with things, and we were able to build something of value. So
clearly,
and when the exit came, there wasn't any kind of, there was no differing views really in terms of whether we should do this or kind of continue to grow the company. So we did have a term sheet also at the same time, so on the table, in order to kind of switch our business from a technology platform into product development. So we were all pretty much aligned as to which direction we wanted to take. So we had a good a good journey with our investors.
Good. If you had the opportunity to go back and give yourself advice on the journey you've been on, what would it be?
I think it would be do your due diligence on the people that you're bringing into the business, and at all levels, and board as well as senior management positions, as well as recruitment, as well as investors. Just do your homework.
Everybody.
I think, yeah.
Yeah. I would wholly concur. I think problems tend to be around people rather than anything else here. Solving problems otherwise. But so, what's the plan then for the future? And moving on from the Synpromics story to your current story with Chromatin Bio, what's the idea behind that, and what's the plan?
Well, as I mentioned before, I love gene regulation. So it's just kind of an opportunity to build a business that's focused on gene regulation, and not just kind of to work with companies in gene therapy to help control their expression of their genes, but to understand how the genome functions and to identify targets within the genome for diseases such as cancer and immune disorders. I think the main difference between what we've done before and Chromatin Bio is that we are purely an informatics company at Chromatin Bio. At Synpromics, we were building promoter libraries, screening those, validating them, and kind of passing the validated sequence on to our partners.
Yep.
We learned an awful lot, doing that over the last decade and a half, and now I don't think we need to build these massive libraries. So what I'm doing with partners now is making a small number of designs that I pass on to them for them to test that in their systems. Because one of the kind of, I wouldn't call it a waste of resources, but one of the kind of aspects that was resource heavy was screening. So we had to develop these cell systems and animal models in order to test our promoters. There's a person that has the best kind of versions of those are our clients.
Right.
So the concept with Chromatin Bio is that we will use our informatics tools and pipelines, identify the optimal sequences, and pass those sequences on to them within a matter of weeks, so that they can get them validated in their own hands with their own models, and then get a quicker answer as to whether or not that's going to be useful for them. And it kind of shortens the timelines, and we get a quicker answer also as to whether our projects are actually going to convert into the downstream milestones. So that's the main difference. And we've also found that Synpromics, a lot of our partners were just asking, can you build us these really powerful promoters? Whereas now partners are coming to us in Chromatin Bio and saying, we need something which is really, really selective to this particular subcell type, this kind of tissue.
Yep.
And we've had to kind of change the way in which we do the analysis in order to accommodate that. So there's also it's very distinguished from the platform we had previously. We've had to kind of gear it towards answering a different type of question.
Has the market generally moved on, in a sense?
I think it has. No one's asking us for more powerful promoters.
Right.
They're saying, we're working with this viral promoter. It gives us the expression we need. So we don't need stronger than that. Just could you make us something which is very specific to this particular cell type? So we spend almost as much time looking at the off-target cells as we do looking at data from the target cells.
Fantastic. Well, I mean, it sounds like you've got another very exciting project on the go, keeping you busy. And listen, thank you very much for your time today. That's been wonderful. Anything further you'd like to add?
No, I think we've covered everything. I mean, thanks very much for having me, and I've really enjoyed kind of going through all of my past experiences.
An absolute pleasure, and a real insight actually into kind of a number of different aspects. I mean, clearly you love the science, and that is, great to see actually, and you still love the science, which after what you've been through is great to see.
No, maybe one thing I would add. So I mean, we we created at Synpromics something of immense value. There was always a vision that this would be of interest to various organizations to incorporate into their kind of platforms. I think at Chromatin Bio, I'm also interested in the business side of things. So I'm interested in creating something which stands on its own 2 feet. Perhaps doesn't necessarily need to go out and fundraise massive amounts. It can actually generate its own revenue whilst kind of enjoying the science and helping patients at the same time.
Very well said. I mean, I think that is a nice ambition, to have a business that in essence is your baby, entirely. Good for you and good luck with it.
Lovely. Thanks. Thanks very much.
Thanks very much.